Welcome to the Fish Labeling Validation Lab. This video will be an overview of the background of the lab as well as an overview of the lab procedures for the lab itself. Fish mislabeling is a big problem in California, both in restaurants and in grocery stores and markets. Here's an example of the high rates of mislabeling in Los Angeles sushi restaurants. In this activity, you will serve as a regulator testing and validating fish from local restaurants to determine whether or not it is properly labeled. In a regular year, you would actually select fish samples yourself from your favorite sushi restaurant or the grocery store, and we would actually test these in class. However, of course, this year we are using some made-up data from some made-up restaurants, but it will really tell us something about how these processes normally go. In order to determine if the fish samples are properly labeled, you'll be doing DNA barcoding analysis. Here are some examples of some different fish species. The top two, A and B, are the same genus, and the bottom two are a different genus. And you can see how the barcodes between similar genus fish are more similar, whereas between the two different genuses they are quite different. Rather than doing whole genome sequencing, we can actually just look at one particular gene, the CoI gene, that has distinct differences between different species of fish, and it can allow us to identify which species of fish we are dealing with. In a regular year, we would do the lab portion of this in class, but it is not something that can really be done at home. So I'm going to give you an overview of the steps of extracting the DNA from the fish and amplifying this particular CoI gene using PCR. The first step is to extract the genomic DNA of the fish that you are using. So students would bring in samples of different fish, we'd make sure everything's labeled. You would cut up a piece of fish, very small, and do a DNA extraction. So using extraction buffers and centrifuges and purifying using a spin column. Once you had extracted and purified the whole genome of the fish DNA, you will want to amplify the specific gene we are interested in. So that's that CoI gene. The way that you amplify a particular section of DNA is using PCR. So PCR is the polymerase chain reaction. It allows you to select a certain part of the DNA and make a whole bunch of copies of it. So we would make a whole bunch of copies of the CoI gene so that we have a large sample to work with. PCR is an incredibly common biotech technique that allows you to make a lot of copies of a section of DNA. In order to do this, you need certain starting materials. You need buffer, you need primers, which are little sections at the start and end of the gene you're interested in. You need DNA polymerase to build the DNA, and you need free nucleotides, so the A's, T's, G's, and C's to build more DNA, in addition to the segment of DNA that you're interested in copying. You combine all of these materials in a thin-walled tube and put them into a thermocycler, which is a temperature regulator that allows you to do this process effectively. The first step is to heat up your sample so the DNA double-stranded helix denatures or separates out into two separate strands. The thermocycler then cools down for long enough for the primers to attach to the beginning and end of the gene you are interested in copying. These are small complementary sequences of DNA that match that starting and ending part of your gene. In the third step, the Taq DNA polymerase, which is an enzyme that's heat resistant, comes in and builds the DNA strand from the free nucleotides you put in to begin with. The cycle then begins all over again with a heating up step where the new strands of DNA denature, separate out, and then more and more strands can be built using the same cycle. Thanks to the Amoeba Sisters for these great graphics. Once you've amplified the CoI gene and made a ton of copies of it, you then run a DNA gel to check and make sure that you actually amplified the right section. The final step is to do the sequencing, which we can't do in our lab, but we can send our samples of amplified CoI gene to a company that will sequence them for us and then send back the A's, T's, G's, and C's, the sequence of results for that gene. The process of sequencing is very similar to the process of PCR with a few key differences, including the inclusion of terminator bases that will both end the DNA strand whenever they are added and contain a fluorescent molecule that is different depending on which base they are. So red versus yellow versus green versus blue for the A, T, G, and C. The same sequence of denaturing, annealing, and elongation will occur, but whenever a terminator base is added, the strand will end there with a fluorescent molecule. In the end, you get thousands upon thousands of different short segments, each ending in a fluorescent terminator base for the different A's, T's, G's, and C's. These are then analyzed using gel electrophoresis, so the smaller fragments will travel farther through the gel, and then you can use fluorescence imagery to see which bases and which size of fragments. The color coding of the fluorescent tags is key here, so if you see a green fluorescent response, you know that corresponds to a particular base, A, T, G, or C. From this information of length combined with base, you can begin to build up the sequence of the sample. Thanks to the Human Genome Project for these graphics. This is where you will be picking up the lab this year. So we have samples of sequences from the CoI gene from different fish that you will be able to analyze using bioinformatics to develop barcodes for these fish. This will allow you to determine what species of fish you are dealing with from your sample. By analyzing your sequencing and bioinformatics results, you will be able to determine if the fish truly is the species it is labeled to be, in this case, fake labels that I made.